Hello, and thank you for joining us. I'm Jasmine Navarez, Digital Content Coordinator here at Simulations Plus, Director of Business Development. Dan O'Connor will introduce our speakers for today. In this webinar, Haiying Zhu, Senior Director of Simulation Technologies, and Neil Miller, Vice President of Simulation Science, will demonstrate how these new features in GastroPlus support research across departments, inform regulatory interactions, and communicate with all software across the Simulation Plus universe. Just a few housekeeping items before we begin. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you're allowing us to contact you for follow-up. You may ask questions via the question panel on your dashboard at any time. We'll address the questions in the Q&A after the presentation. Take it away, Dan.
Well, good morning, good afternoon, or wherever you are in the world. Welcome to 2023. Can you believe that it is already January 2023? But we have some great news to share this morning with the new introduction of 9.8.3. There's a slick name if I ever heard one. We have two of our principal scientists who will be presenting today. Neil Miller, whom you know is Vice President for Simulation Sciences. He's located in UK. And then his colleague, Haiying Zhu, who's a Senior Director of Simulation Technologies, who's really the driving force behind a lot of the development and creation of the advances within the GastroPlus platform. We'll go for about 20-25 minutes of presentation that will be followed by a Q&A session. So please do feel free to send your questions to us in real time, and we'll handle as many of those as we can during the conduct of this call, followed by afterwards trying to get back to you if you have any follow-up. So without further ado, may I introduce Neil Miller, who will take us forward in the next phase of our presentation. Neil?
Thanks, Dan. Before I say a few words, I think we're going to go for a quick poll question.
Yes. So our first poll is, where are you in your GastroPlus journey? Are you a hatchling, nestling, or fledgling? And then we'll take a few seconds to answer those. See them coming in. All right. So it looks like we have hatchling and fledgling and some nestling. So we have beginners and experts and intermediate as well.
Thanks, Jasmine. Looks like we've got a nice mixture there, almost an even split. So as Jasmine and Dan said, I'm Neil Miller, the VP for Simulation Sciences. So I oversee the application of GastroPlus to real-world problems, and also the development of GastroPlus to drive model-based drug development in all phases of drug discovery, translational research, and clinical development. It is my absolute pleasure to introduce you to Haiying Zhu, Senior Director for Simulation Technologies, who is the driving force behind version 9.8.3. Now, Haiying has been with Simulations Plus for coming up to 14 years and is one of our leading GastroPlus trainers. She works on grants, consulting studies, publications, customer support, as well as software development. So if you want to know anything about GastroPlus, then just ask Haiying. So please take it away, Haiying.
Thank you. Thank you, Neil and Dan, for the introduction. And also, good evening, good morning, good afternoon, everyone. So for today's webinar, so I will talk about what's new in GastroPlus version 9.8.3. Yeah, sorry. Yeah. This slide will show you some of the main changes we made in GastroPlus 9.8.3. So I divided them on the different modules. For the ACAT model, we increased the flexibility in defined suspension dosing. For the PBPK plus module, we added extra disease population, the NAFL and NASH disease population. And also we added the swine PBPK model. For the additional dosage routes module, for the ocular, we add the melanosom trapping for the ocular. For the oral cavity module, you're able to add mix multiple dose for the oral cavity dosage form. So these are some like enhancement in the additional dosage route. For the compartment PK model, we add the option to synchronize liver first pass extraction during the optimization of liver clearance. For ADIM predictor module, we add the mouse specific property prediction model. We also add some new enzyme transporter distribution in the metabolism and the transporter model. For population simulator, you're able to see the population simulation results in the Monolix compatible like format, what the Monolix required, the input format. So you could link the GastroPlus with Monolix. There are also some other small changes. Here I list one example, like for the default output format for PSA, parameter sensitivity analysis, and population simulation, that will be Excel format. So this will be set as our default output. But you will still have like the default output, like the population simulation, like like the population result. That will still have that. You could reload it for the simulation. We still have that, but we also add the Excel like a format as a default. First, I want to talk about the suspension dosing. So in GastroPlus, if you use the suspension dosing, now you have extra like settings for the suspension vehicles. If you click on this button, like the suspension solubility, this button, and you will open a new window. So in this window, you can set suspension vehicle solubility setting. In the previous release version, you can set solubility in dose vehicle and also the vehicle volume per dose. So based on this information, the program will automatically calculate the percent of the dose already being dissolved in the vehicle, and also the percent of dose still as solid undissolved one. So during your simulation, these two parts will be used. So one is already dissolved, one is undissolved. You do not need to set as mixed multiple dose, like part as dissolved, part as undissolved. Now the program will automatically take care of it based on your vehicle, like the solubility setting. So our users really like this function, but they also provide extra requests because during the early formulation development, sometimes they use constant suspension volume. So in order to make it more easy for the user, we add an option. So you could select a constant suspension volume. So the user do not need to manually calculate what will be the vehicle volume per dose. Now I just set what will be my suspension volume I want to use. So with this one, I show you an example. Like if we use like two different dose, like one is dose level is 100 milligram, another one is at 200 milligram. If we do not use constant suspension volume, we use like a vehicle volume per dose. So that's a constant. So the solubility in dose as like in dose vehicle as 1 milligram per milliliter, and we use 0.1 like milliliter per milligram for vehicle volume per dose. And with 100 milligram, we have 10% of the dose already dissolved, and 19% still as solid undissolved. For the same setting, if we set the dose to 200 milligrams, that will be the same because the vehicle volume will increase. So we still have like the percent of dose as dissolved as 10%, and the percent of dose as solid undissolved as 90%. But if we select a constant suspension volume, so now the vehicle volume will be a constant. So we change the setting like now this input window will represent what's the vehicle volume you want to use. If we use 10 for these two different dose levels, Now the percent of the, for 100 milligrams, the percent of dose as dissolved is 10%. And for this 200 milligrams, the percent as dissolved of the dose is 5%. 95 is as undissolved. So we add this option, so now user could have more flexibility, like to define the suspension dosage form. Yeah. The next item I want to talk about is the improvement in PBPK plus module. And we expand the disease population and try to incorporate like the NASH and NAFLD disease population. And for this one, like for the disease-related change, like the NAFLD and NASH population, so it do affect like the different mechanisms related to the drug absorption and the distribution. It affects like the absorption, and it affects the distribution and the renal clearance, the different elimination, like in the liver. For the absorption part, it's really like for the patient with obesity or like the type 2 diabetes, they show like highly viable and also some delay in the gastric emptying. And just due to the dysbiosis in the gut, this may also affect the enterohepatic circulation of the drug and also bile acid. Currently, we didn't have like the protein level of the data about the CYP enzyme and also the drug transporter in the intestinal, the expression. For the distribution, most of the like the subjects, the patients, they do show like the higher, like so they do show the higher body weight and the BMI, and but still around 25% of the NAFLD patients, so they have normal BMI, so we call it like a lean, like a NAFLD patient. Generally, these patients will have a higher like the total body and the liver fat, and also the cardiac output could like increase by 17%. And for some of the protein, like the plasma, glycoprotein, the albumin, and the hepatocyte, they keep like the level as a healthy subject. For the renal secretion, we do say like the GFR decrease, and also the transporter, we can find like they could change the different ways, some increase, some decrease. And for the hepatic skill factor decrease by like 20%, and also the liver weight increase, but its blood flow decrease. And we also see like the change in the CYP enzyme in different directions, the non-CYP enzyme, and also the transporters. So all this information will be incorporated in the virtual, like the PBPK model for the NAFLD and the NASH patient. And also depends on like the different stage of the disease, and we separate them to NAFLD and also NASH patients. We separate them to two, like the groups. And this is we take from Jeffrey's presentation at the 2022 IS conference, and he mainly focused on the validation of the PBPK model in GastroPlus about the NAFLD and the NASH patients. Yeah, and this slide I just want to show you like within like the GastroPlus and the actual two groups of the disease we added, and except the cirrhosis. So we already added in, like we already have it in the previous released version. Now we have two like extra groups. One is simple cirrhosis. So we call it NAFL, like the disease population, which only have the cirrhosis, but do not have any inflammation of the fibrosis of the in the patient. And another one is the NASH. So So this is still hepatitis, like this patient group. So this will be the two disease population we add. In this slide, I show you three, like the typical subject. One is a healthy subject, another is for the simple steatosis, we call it a NAFL patient, and another one is a NASH patient. I just show you in the liver some of the physiological parameters change. Definitely there are more change like in other like tissues and in the whole body physiology. So for this one, I just show you the liver change. And then we can find even though the patient have the same, the subject have the same body weight, and the liver volume, like we can find it's increase in the NASH subject, and also the blood flow decrease as the development of the disease at the different stage of the disease. Also, the lower part of this shows the enzyme, CYP enzyme change. And for this 2E1, we can find like the expression like increase and in the NASH population subject, and also for 3A4, 3A5, the expression decrease. So they do like the enzyme like change in different ways. This slide shows in the NAFLD disease population about some changes in kidney. And also like the first, like the subject represents a healthy subject, and then the NAFL and also NASH subject. The blood flow, we can find its increase in the kidney at the different stage of the disease. And the GFR, we can find its decrease in the NASH subject. And in this graph, we also show you the MRP3, the transporter change. And we can find the expression level decrease when compared with the healthy subject and with the disease population. Yeah, so this just to show you some example about the physiology we incorporated in liver and kidney. So there are more changes in the PBPK model for this disease population. From Jeffrey's presentation, like at the ISS conference, he also showed several compounds for the validation of the model of this NASH and NAFLD disease population. In today's webinar, I will show you one example, is the pioglitazole example. And this is shows the metabolic pathway of this compound. And in the PBPK model, we consider the hydroxy pioglitazole as the major primary active metabolite. So we consider the parent compound and also the metabolite. And also the formation from the parent to metabolite is mainly like cleared metabolite by CYP2C8. And so we will use CYP2C8 to describe the clearance of pioglitazole. For the hydroxy pioglitazole, we also incorporate in the model, we created the PBPK model for it. But its clearance, and since we do not have enough like the information, we just assume a linear clearance in the liver for the metabolite. That will be some of the settings related with this compound. For this, we found from the literature, there are some experimental data, like clinical data in this healthy subject, so non-NAFLD, non-diabetic subjects. And in the study, they have 26 subjects, and also the mean age is 29 years old. The mean body weight is 67 kilograms. So the dose is multiple dose, is 30 milligrams per day for 8 days. And in our simulation, we also do the population simulation. We match the age, match the body weight. And this is the simulation result. The blue line shows the mean simulated concentration profile for the pioglitazole. And this green, like the space shows 5%, so 5 to 95 percentile of the prediction of the simulation. So if we look only on the day 8, it has observed the data. If we take the last dose, and we can see the model prediction have a great match to the observed data. This is for the parent compound. And for the metabolite, hydroxypygalactone, and there is accumulation for this metabolite, and also it's already reached steady state at day 8. If we say only the last dose, and we could find like the model to able to give good simulation of also the metabolite. Now we use the same model. We try to simulate the exposure in NASH patients. So all the physiological change already be incorporated in the NASH PBPK model. And from the clinical data from the literature, for the NASH patients, they have 56 subjects, and also the age is the mean age is 54 years old. The BMI of the body weight is around 34 BMI. The dose level is 45 milligrams per day, so it's have some different compared to the healthy subjects. So in the disease population, they use 45 milligrams per day. So this simulation, so we also simulated 8 days, it's reached steady state. And for this observed data, they only measure the steady state minimal concentration. So they didn't measure the whole profile. So what we try to compare is the steady state, the minimal concentration. And this is from the last dose and the minimal concentration. And also the blue line shows the simulated mean profile. And then the gray area shows the 5 to 95 percentile of the simulation. So we also try to match the NASH patient, like the age, the BMI, all this information. So on the right side, it shows the log scale, compare the observed and then the simulated data. Now we look at for this NASH patient for this metabolite hydroxypygalactone, and we can see the prediction, also the C minimal, at the steady state, the C minimal prediction match the observed data, and the blue line shows the simulated the mean profile, and this is a log scale. Yeah, so from this example, so we do say the default, the physiological change we incorporate in NASH patients do able to explain the exposure difference for pyrgalactone and also its metabolite in the disease population. So in Jeffrey's poster, there are also more validation examples. If you are interested, you could check it. Now, this slide we try to compare what's the normalized the ratio of this steady state and minimal concentration difference between the healthy and the NASH population. For the pyrgalactone, for the parent compound, we can find for the healthy subject for 30 milligrams per kilogram. So the clinical observed data, the minimal concentration at the steady state is 140, and the model predicted it's 141. It's very close. And for the NASH, the population, this is 45 milligrams per day, and also the observed data and also model prediction are very consistent. If we normalize this to the dose, and then we can find the say minimal it's increased in the disease population by almost 40, more than 40% increase. For the observed, say the normalized ratio between the disease NASH population and the healthy population is 1.46, and the predicted is 1.42. It's very consistent. And if we look at the metabolite, And we also look at the dose normalized the minimal steady state minimal concentration ratio, and we can find there are decrease in the metabolite concentration. So for the clinical observed, and it's decreased by almost 32%. So the disease population compared with the non-disease population healthy, like it's 0.68. Yeah, the predicted is 0.66. So the decrease in the hydroxypyrglutatone, the minimal concentration, so it's also consistent between the clinical observed and also the model prediction. The next improvement in the PBPK model, we add the swine, the PBPK model. So for this swine PBPK model, this is mainly really like to use for like the food animal. So we try to also have some collaboration project, try to able to predict the drug concentration in food animals. So the first model we add is the swine model. We still have other like animal in development. So this is mainly like because the PBPK model is really important. It's very useful to predict like especially like for the drug with off-label use, or if it's with like changes or varying like dose regimen. Because to consider the food safety, we do need to know like the time like the animal need for the drug or chemical clearance following the exposure. So we could assume the residual like in the food animal or like in specific tissue, like it will below the safety level for the consumption. So that's why we have the collaboration like to develop the food animals. So for this swine model, and you will find you are also able to set age. Like if you select the species to swine, you're also able to set age. If you are already a user like of GastroPlus, you will find for other like animal PBPK model, like for rat, for mouse, for dog, the model we incorporate in GastroPlus, it's adult animal. So you are not able to set the age. It always represents the adult. But for swine model, you are able to set age because the different age, they will have different like gut physiology, and they will also have different like the PBPK model, the whole body. This example on this slide I show you is a swine 250 days old or compared with 180 days old. So the average, like the mean, like the body weight, so there are difference, and also the cardiac output has different. Also, like for the different age, like of the swine model, they also have different ACAT like model. For this one, they all like under fasted condition. If we compare 250 like days old compared with 180 days old, we can find along the GI tract, the length and also the radius of each like a GI segment are different. So these are all incorporated in the model to account for the age difference in the swine. Also, you could set like different the fasted and fed condition. We do incorporate the physiological based fasted ACAT model and also the fed state ACAT model. And for this one, you can see the fasted condition and also the fed condition. They will have a specific different pH, transient time, also the volume. So the stomach volume you could use to represent that fed condition. So we already incorporate all this in the ACAT model for swine. So this slide also show you, our collaborator, Rob Dvorsky, also present some validation of the swine model at the 2022 SOT conference. So in the poster, in the project, they do try to explore like the new platform, like try to combine our ADIM predictor with GastroPlus swine PBPK model. Because for the food animals, for some of the like the compounds, they do have very limited, like very sparse data about the model like parameters. So that's why they want to explore the combination of the ADIM predictor with the like the GastroPlus, the PBPK model. For this in example, they show penicillin, and they only use the ADIM predictor to predict all the drug properties, like all the parameters needed for the model. What they need is take from the literature, from the Lee publication in 2017 about the kidney urine secretion and also the partitioning, the tissue partitioning of the kidney. So these are two parameters they take from the literature, and then they use the swine model from the GastroPlus to build the model. And this example shows like for this, this will be 15 milligram per kilogram intramuscular injection for 3 times. And the blue line shows the concentration in plasma. The green line shows the concentration in kidney tissue. So we could say like it's give like a good prediction of like both in the plasma and also in the tissue. It will be very helpful to help to define like when, so what will be the exposure, like the safe withdrawal interval, like what will be the time required for the animal to clear the compound, and then the concentration will reach like a safe level, so able to be consumed. Yeah. So on the right side, it shows 65 milligram per kilogram penicillin. So this dose 5 times, also for intramuscular injections. And we can see the same model also predicts a very good prediction for the different dose level in both the plasma and also in the kidney tissue. The next I want to talk about is some improvement in the additional dosage route. So in additional dosage route, first I want to talk about the improvement in the ocular. So we add the melanosomes trapping in the ocular. So this is mainly because we have a collaboration project that try to validate our current ocular platform, like absorption platform, and also try to improve the gaps between like from the animal data, try to predict the human the eye exposure. So we try to improve our ocular like the absorption model. This is one of the improvements we add is add the melanosome trapping. So for this one, so for some of the like the tissue, this is our ocular, like the current, we divide them to multiple like the subcompartment, and for some of the tissue, they will have melanosome compartment, and if you click on one of these like the compartment, you can see some of the parameters related to the melanosome like compartment, and what is the pH and what is the volume fraction. And being able to calculate the partitioning in the melanosome like compartment, we also have some like eye tissue parameters. It's tissue composition, like it's neutral lipid and all this content, and also what's the plasma, all this is composition, red blood cell composition. So lots of this is already defined in the PBPK model. You don't need to worry about where I could get this data. So it's already defined, and it's also be used for the PBPK model, the tissue KP calculation. And with this, the physiological parameters, and then we based on some assumptions, like outside of the melanosome. So we consider the binding to lipid. So this will be similar as our PBPK tissue, like KP, the binding, the KP calculation. So we will take the similar, treat it similar as the general PBPK KP calculation. And for the melanosome, so we consider the binding, only the binding to the melanin. So this will be some of the assumptions we consider to calculate the binding in the outside the melanosome in the eye tissue, and also what will be the binding in the melanosome. And we can calculate the KP between the tissue, total tissue, and the melanosome. This is an example I want to show you. It shows like why the importance we add the melanosome trapping. And this example is so this is the pimolonol, like the solution. So for this one, it shows generally it's really good, like the prediction, like for the concentration, for the compound concentration in different location of the eye. But for some of the tissue, it do shows like we underpredict the exposure. This example I want to show you is in ICB. So the model do predict like we underpredict the Cmax, and we also underpredict like the whole exposure. If we consider the melanosomal trapping, and if we do not consider melanin binding, we only consider, okay, the difference is because of the pH within the melanosome compartment, because the low pH is cause different ionization. So if we only consider the ionization difference, and that it's able to, the melanosome trapping is able like to help to describe the Cmax. The Cmax do got some improvement, but still we couldn't explain the lung exposure like in this tissue. So if we consider the melanin binding within the melanosome like compartment, we also consider its pH. So we consider both. We also consider its pH like difference. We consider both mechanisms. Now we can find the prediction like in this tissue is really improved. It's able to explain the lung exposure in the ICD. So this we add this mechanism is really based on like our some validation examples. We do shows like in some tissue for a certain compound, they shows lung exposure. This should be contributed to actual trapping in the melanosome trapping in the eye tissue. So that's why we incorporate this, could give us better prediction in the eye. For the compartment PK, we add the option to synchronize the liver first pass extraction when try to optimize the liver clearance. And if we know like our compartment model, you're able to set the first pass extraction, also to set the clearance, general clearance. So these two could be optimized. If you want to optimize these parameters, it could be set as independent, like two parameters during optimization. But actually in real like situation, these two are correlated. So if you have a higher like liver clearance, and then you could have a higher like first pass extraction. So that's why we add an option. So when you try to optimize clearance, the program will automatically update the first pass extraction. So you do not need to consider them as two separate parameters. So if you, so this is during like the optimization parameter selection. If you select this synchronized first pass extraction and then clearance, now this first pass liver first pass extraction will be disabled because this will be automatically synchronized with the clearance. So this will be updated at each optimization step. So finally, you will get both the liver first pass extraction and updated based on the liver clearance, the optimized liver clearance. For the aging predictor module, we add the most predictive prediction model. So in our previous released version, when you try to import the structure and the model could, the aging predictor module could predict like the most, some of the model parameters related like to rat and in human for you, like RBP, FUP, and also the clearance. Now we also add the model for mouse. When you import the structure, if you select a mouse model, and then the program will also predict like the mouse RBP, the mouse FUP, and our clearance. At the node area, like the prediction for human RBP, the prediction for like human and rat, like all this RBP, FUP, and also clearance will show in the node area. So when you try to use the in silico property, but you want to simulate the rat and make sure you updated this like a prediction from the rat model. Yeah, but so in this released version, we add the mouse prediction model. You do not need to borrow the value from other species prediction. For the population simulator, and we provided an extra selection like for the extra simulation mode. You could simulate like a Monolix, like the compatible like the mode. So this will be generate the Monolix compatible output file. You could select this from the main GastroPlus simulation mode selection, or you could through the menu items. And so from the simulation setup, you have the mode, and you could simulate the Monolix. So this mode. So it will run the population simulation, but the output, you could have an extra output file that will be Monolix like compatible output file. This table shows you an example of the output, and the variables name are saved in the specific requirement following the Monolix requirement. And you could further use Monolix to analyze the data, the output file. For the metabolism and the transporter module, we add some new enzyme expression. So we add the long-term microsome protein expression for mouse, swine, mini pig, and rabbit. And so in their enzyme table, in the liver, you could find there is an enzyme named long-term microsome protein. And in human gut, we also add 286 expression. The 286 has very minimal expression along the human GI tract, so it's all 0. So we add the expression. We also updated the 2D6 pro-metabolizer expression level. So in our PBPK model, we do add like for the 2D6, we have the ultra metabolizer, extensive metabolizer, intermediate metabolizer, and also poor metabolizer. For poor metabolizer expression, we have been updated in this version. And for the validation of this, there is an example to show you like a validation of this updated poor metabolizer expression. So I took this from one of the manuscripts still in preparation from Maxim. So this is a metoprolol example. So during the baseline, the model development, and we have the IV data help us to define its the clearance and also its distribution. Based on this model developed using the IV infusion, and then we try to predict what will be the RR tablet, the oral dose, and in 3 groups. So they have ultra metabolizer and also extensive metabolizer and poor metabolizer. The green line shows the ultra metabolizer. Orange line shows the prediction from the extensive metabolizer. Blue line shows the prediction from the poor metabolizer. And all these dots represent observed data. We could find, and the default expression we add for 2D6 to able to describe the 2 different groups with different the 2D6, the expression level we add as a default. I think that would be some of the main change I want to describe in this webinar. Definitely there are also some small changes as I mentioned like the default expression, default output format for this, the PSA and the population simulations. So there are also some minor changes. Today, I just talk about some of the major changes we made in this version. And also at the end, I want to thank the different developer team from Simulations Plus and the simulation technology team. We have Jim Mullin and Jessica, Daniela, Manash, and so Priyati also contributed to this version. We thanks Maxim to help the improvement in the ocular module and also the validation examples. Thank Ola to help us to do lots of the testing. Also special support from Neil Miller and also Vera. I also want to thank lots of the useful suggestions from our users. Yeah. Thank you for your time and attention. So I think it's time to take some questions.
So before we do the Q&A, I do have one more poll for everybody to answer. And the question is, how quickly will you start using GastroPlus for your modeling? And we'll give everybody a few seconds to respond. Awesome, I see most of them coming in. So almost, again, a good mix of everything. I'll pass things back to you.
Thank you very much, Jasmine. I'd like to see the fact there that I'm on it came out top, only slightly, but 34% of people are going to use it straight away. So that's good news. We now open it up for questions, Q&A. We do have one question at the moment, or there might be another one just coming in. But if you would like to see, and if we do have time, some of the functionality demoed live in the time that we have left, please post that in the chat as well. So the first question that we've got is there a feasibility to predict CSF concentrations in different species from one? So currently, we don't have CSF included in the PBPK model. We do have ideas about potential models, but there's been a lack of resource at this point to include it yet. So it has been thought about. And if I could sort of advertise at this point, if you do have questions about GastroPlus when we're not holding webinars, a good font of information is our resource center on our website. So if you go to the resource center there, you can type in search terms and that will bring up posters, publications, webinars to give you a lot more information about GastroPlus and our other products and how they have been applied. So that's the first question. Let's see if there's another one come in. Not in the Q&A. Okay, so the next question is in the chat. So it is it possible to use the program with metal containing compounds? So Haiying, are you aware of GastroPlus being used to predict using for compounds with that contain metal?
Yeah. Okay, I saw some like the user already answered it is yes. Yeah. Yeah, personally, I do not have experience. Yeah, so I may need to go back to check but I do see like some answers, like some user mentioned like, yes, they do use it. Yeah, I
think it could be done, but we'd have to get the predictions from ADMET Predictor or all the properties, because ADMET Predictor won't contain them. So it could be done, the platform's there, but there might be some issues on some of the parameters.
Hey, Neil, this is Dan. Just might be a good time to mention the GastroPlus user group, where an awful lot of questions back and forth and strategies for implementation are shared.
Thank you, Dan. That is a good advert. So there is a LinkedIn page there where there is an opportunity for users to post questions to get answers from other users, because obviously we are not aware how GastroPlus has been applied in every organization. And so you can pull on the experience and the knowledge of other users via the GastroPlus user group, and specifically their LinkedIn webpage, where there is a Q&A section. So I think we've got another question here coming in from Bart, and this is around the suspension dosing. So asking for more clarity around addressing the undissolved fraction and the dissolved fraction as a suspension. So it's still not clear how to address that. So I'm not quite sure if that's how you would enter it in the program or how the program's handling it.
So you said, could you repeat the question? Yeah.
Sure. The question is, it is still not clear for me how to address the undissolved fraction and the dissolved fraction in a suspension.
Yeah, I think currently, like with the suspension, like the settings, you need to provide like your compound solubility in the suspension vehicle. And also your suspension vehicle, like the volume, like per dose, or if you use like constant volume, then the suspension volume you want to use for your suspension like formulation. Then the program will calculate it. Okay, the amount already dissolved based on the solubility of your compound in the vehicles, and try to calculate what's the dissolved amount, the fraction. And then the leftover will be undissolved. So that means during simulation, it will follow either like the Johnson method or other method you define, like Z factors, and they will start to dissolve. So this is like the program will use the solubility of your compound in the suspension vehicle, and also the vehicle volume, try to calculate it. Yeah.
Thanks, Heidi. So the software itself actually calculates the amount that's in solution based on the inputs, the solubility and the volume, etc. In the past, I think you mentioned it during the presentation, Heidi, that people would have to use an MDD file to kind of specify those, but now the software is calculating that automatically. So the next question we've got is, if the clearance is going to be optimized by turning on sync fit, can we also be able to see the optimized percent FPE?
Yes. So after your optimization of the when you try to optimize clearance, so the I couldn't say it's optimized the first pass extraction, because now the liver first pass extraction will not be available for optimization, because they are not treated as like two independent parameters. So that means you optimize your clearance. Each step you get a clearance value, and then it will automatically update what will be the corresponding liver first pass extraction. So if you really want to, okay, optimize like the first pass extraction as a separate like value, you may want to like optimize other, like optimize first pass extraction from gut, because now the liver first pass extraction, excuse me, will be synchronized with the with the general clearance or the liver clearance. Excuse me. Yeah.
Okay, thank you, Heidi. The next question is, do you actually need an active GastroPlus license to attend any of our training courses? And the answer is no. When you sign up to those training courses, you do get access to the software to be able to carry out the hands-on exercises which complement the lectures within those courses. So no, you don't need an active license to attend our courses.
Neil, if I may, the complementary training sessions you do. So if
yes, thank you.
Yeah, if it's an advanced GastroPlus training, what we would call a paid training, The license is part of that package. If you're coming for introductory, complimentary training, we would expect you to have access either to a trial license or to an active licensing within your own organization.
Thank you for the clarity, then. The question I was reading was, in fact, for the Advanced GastroPlus course. So thank you for the clarity that for those complimentary sessions, you do need a GastroPlus license. Which kind of links onto or follows on to the next question, which is asking around finding out about more information. Is there any instructions or guidelines, PDFs that we can use to learn more about GastroPlus? Well, there's the complimentary sessions that Dan's mentioned for some live hands-on training, but we do have tutorials with the software as well. So when you fire up the software, when you start up the software, you can quickly open up tutorials which demonstrate, highlight, showcase all of the functionality of the software. So within the PDF, the manual PDF, you can access the manual via the software itself live, but also within your install, there is a PDF of the manual as well. And there's step-by-step instructions for each of the tutorials so that you can train yourself in any of the functionality that you have access to within the software.
Yeah, in addition, this is Dan again. In addition, we do have a library of web-based videos that highlight a lot of the various areas of functionality for GastroPlus, and certainly at any time, we would be more than happy to make a presentation in any subject area for a prospective client or a current user, especially as current users are considering adding additional modules or connecting, as we mentioned, to Monolix suite. We can help with respect to providing information on any of those advances.
Thanks, Dan. I think we've got time for one or two more questions. The next question that's come in is that we have seen some further adjustments in version 9.8.3, such as an update in the kidney tubular volume in advanced settings. My question is, what is the expected deviation between the earlier version of GastroPlus and 9.8.3 for the similar record, or what is the accepted deviation?
Yeah, so actually, yeah, you are correct. So, but this kidney tubular volume, so it's already changing like in 2000.8.2 versions. It's not a new change in 9.8.3 version. It's already like has been changed in previous version. If you compare like version 9.7 with the 9.8.3, you may find it has been changed, but it's already been changed like introduced this change in previous version. So we incorporated this change because we have like some like other project from like other departments. We find our current like the volume setting is a little bit small, so we need to update it with the new like the values. So that's why we did. And yeah, I would say for some of the compounds, there might be some difference, like cause some difference in the simulation result. Like I would say it really depends on how much is the difference. We might need to update the model, because for some of the compound, most of the compound, I would say the influence might be minimal. And if we do say like a significant difference, and we may want to like either update the model, or we try to use the like the old the previous version, the PBPK model, instead of like use the new model, because your data already like able to the model from previous like database able to explain all your data. I would suggest so one suggestion is, if you really want to use the new like the physiological model, PBPK model, you may want to update the model, or if you could use the PBPK model built from previous version of GastroPlus.
Following on from that, I think it's worth mentioning that we're always reviewing the literature, and if we find new information to update the models, we will do that. it's probably also worth mentioning that if you open up an old model within the software, you should get the same results. It's only when you're creating the model with the new version that you would get, for example, the newer kidney tubule volume. If you opened up an old model, you get the same results because it would be using the same input parameters. So I'm looking at the time, and unfortunately, we are at time, and I need to hand back to Jasmine to close out for today. But we will make a note of those questions that we haven't been able to answer, and we will try to get back to those individuals. So thank you for your time, and I'll hand over to Jasmine to close us out today.
Yes, thank you, Neil. Thank you, Haiying. Thank you to all who attended. For any questions we didn't get to, we can also answer them on social media. We invite our audience to learn more about GastroPlus or other products and services on our website, as well as our University Plus program, which offers 1-year access to Simulations Plus software to students and educators at accredited universities worldwide. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you so much. We'll see you next time.